Hello, everyone. My name is Anna Lee. I'm a research assistant at Kennedy Krieger Institute. Today, I'm here to talk to you about in vivo interstitial and cerebral spinal fluid exchanging process detected by non-invasive magnetization transfer indirect spin labeling MRI. The study of CSF has begun almost a century ago, but it has drawn much attention recently because of the discovery of the lymphatic system back in 2013. It was believed that a portion of the subarachnoid CSF recirculates through the brain parenchyma and exchanges with the ISF and then flow back to CSF via the lymphatic system. It is also shown that the lymphatic system has a strong correlation with aging and other neural diseases. Therefore, it's no surprise that people want to understand the circulation and production of CSF. There are many MRI techniques developed for imaging the lymphatic system. However, many of them can only measure CSF in bulk. Therefore, oftentimes a contrasting agent is needed because the CSF flow can be relatively slow. Here is an example that uses a gadolinium-based contrasting agent. It is infused intracisternally, and it can also be done intracranially, both of which are highly invasive. And in the back of our mind, we still worry about the side effect of residual gadolinium tracer in patients' brain. Recently, D-glucose as a natural biodegradable tracer was proposed and became available. Here is a research from our institute that study Alzheimer's disease through the glucose uptake. Although contrasting agent has been applied in human study, it is far ideal for routine and repeated measurements on patients for the obvious reason. A non-invasive method is in demand. We are here to propose a non-invasive and translational MRI technique that we developed to measure the water exchange at the interface between the brain parenchyma and the ventricle. The method is called magnetization transfer indirect spin labeling, MISL. Before I tell you more about MISL, I want to point out what MISL measures. Here is the cartoon version of the interface between the brain parenchyma and the ventricle. Bridging them is a line of ependymal cells. There are two flows coming into the ventricle. One is from the brain parenchyma to the ventricles, ISF, CSF flow. The other one is from the ependymal cell to the ventricles, ependyma, CSF flows. MISL measures the combination of both flows. And we believe that the ependyma to CSF flow is the major contributor to MISL. MISL takes advantage of the different protein concentration between brain parenchyma and CSF, being that brain parenchyma have 200 times more micromolecules than CSF. So when a pulse is applied to label micromolecule in the brain, the parenchyma is labeled, but the CSF is left intact because there's only a trace amount of micromolecule in CSF. And in the parenchyma, in this figure, we separate the micromolecules from the water molecules for better demonstration, but in reality, they're mixed together. The labeled micromolecules are going to rapidly exchange proton with the water molecule. And in this way, the water is indirectly labeled through magnetization transfer. And the labeled water molecule are going to travel to CSF/ventricle and cause a signal change as CSF. There are two sequences for MISL. The first one is Pulsed-MISL, which is a pulse followed by post-labeling delay. This is a mixing time before the acquisition. And then the second sequence is Continuous-MISL. This sequence is identical to the conventional pulse and continuous wave CEST sequence, except you can insert post-labeling delay before acquisition if you want. So to study CSF, the first step is to study the distribution of the ventricles, and that was achieved through long TE-TSE images. And to make sure that the region of interest we pick, fourth ventricle perivascular space, lateral ventricle, third ventricle, do not overlap with brain. With blood vessel, we overlap the long TE-TSE images with graphic maps. And next, we validated MISL with extracted CSF. So here is a healthy, wild-type mouse, and we use the needle to extract fresh CSF from the mouse ventricles. And a continuous MISL sequence is applied to this extracted CSF. And what we observed is the strong water signal at 0 ppm, but anything else is at the baseline. And the same sequence was applied to a mouse brain. This is in the physiological setting. So the brain prochema has a strong signal, which is what we expected, because the prochema is the part we labeled. And we were very happy to see that the signal of CSF is no longer at the baseline because there's water exchange from the brain prochema to the CSF. So these two figures really show that the isolated CSF does not cause any signal change. Only the water traveling from the prochema to the ventricle can cause signal changes in the brain CSF, which shows that MISL works. So for post-MISL, a pulse is applied, followed by PLD ranging from 0 to 4 seconds. This is to study the kinetics of the flow. And both lateral ventricle and third ventricle peaked at 1.5 seconds. And from the shape of the curve, we were able to calculate the bolus duration as 0.7 seconds. And from this, we speculated that the majority of the signal comes from the ependyma to the ventricle because of the shortness of this bolus duration. Because if the signal were dominated by a flow coming from the inner prochema to the ventricle, then we would expect this bolus duration to be much longer. Continuous-MISL applies a train of pulses to increase the labeling efficiency and to obtain images with better resolution. Here's the result we found. And what we learned is that water molecule exchanges at different rate and different quantity at different sites in the brain. For example, the perivascular space has the lowest amount of water exchange, and the fourth ventricle has the highest amount of water exchange. And last, we used MISL to study age and AQP4 dependent CSF-ISF exchange. Here's the result. So for the third ventricle, young mice preferred better than adult and AQP4 knockout mice. Perivascular space, they're pretty much all the same. And for the fourth ventricle, young mice performed again better than the rest. For the fourth ventricle, it is higher than the rest of the ventricle in perivascular space. And again, young mice performed better. So the conclusion we made from this is the process we measured is more age dependent and not so much AQP4 dependent, which further validate our proposed earlier that MISL is dominated by the flow from ependyma to the ventricle because this process is not AQP4 dependent. So in short, MISL is able to measure the combination of flow from ependyma and ventricle, ependyma to the ventricle and the prochema to the ventricle, and the ependyma to the ventricle being the major contributor. We also have follow-up study that can measure the water exchange from the ventricle to the prochema and ependyma. And we would be happy to present that in the future. Last, I'd like to thank my collaborator from Johns Hopkins University Kennedy Krieger Institute, Massachusetts General Hospital from the United States, and Karolinska Institute from Sweden. And thank you very much for your time and attention.